Hello, biologists. Welcome to the last lecture of the term. Today, we are going to review for the final together. So very exciting. My plan for this is I'm going to show you some questions, and then I'm going to give you 5 seconds to answer it yourself. And if you need more time, just pause the video. I don't want to give you too long because then we'll just be sitting here in silence and it'll be weird. So if 5 seconds seems really short, please pause it, think about it, and then hit play again. I do want you to practice answering these questions on your own. So don't just listen to me go through the answers, which I will, but I want you to really give it a go. And if there's any questions that you know you can't answer, that's a good indicator that this might be a subject or one of the topics that you want to study again. So let's get started. Which of these is the smallest? A bacteria, B, a chromosome, C, a nucleotide, D, a genome, or E, a gene? If you said a nucleotide, you are correct. So nucleotides are what DNA is composed of. A piece of DNA that codes for a protein is called a gene. So nucleotide, then gene. Genes are located on chromosomes, so chromosome is bigger than gene. All of your chromosomes together are your genome, that's your genetic material. And then if you're a bacteria, you have a genome. So bacteria is biggest and nucleotide is smallest. In a DNA molecule that is made of 20% adenine nucleotides, which percentage would be made of guanine nucleotides? If you said 30, you are correct. So 20% are A. A always binds with T. So that's got to be 20% as well, which means that 100 minus 40 is 60. 60% remaining is made of C's and G's. So you divide that 60% by 2, leaving you with 30% are G's. The MYBPC3 gene causes hypertrophic cardiomyopathy. This is the leading cause of death in young athletes. What are the subunits that would make up the MYBPC3 gene? A gene is a piece of DNA, so the subunits of DNA are nucleotides. What are the 2 copies of a chromosome connected at the centromere called? And we have an image there. You can see the centromere is in the center, and it's 2 identical copies of a chromosome connected. If you said sister chromatids, you are correct. These are identical chromosomes attached to the centromere. That's what we call sister chromatids. Which of these, A, B, or C, shows homologous chromosomes? B is the centromere. We saw that labeled on the last image. One of those is a sister chromatid. So C, the purple and the green, are both sister chromatids. That means that A is homologous chromosomes. What's happening here is you have one set from mom, one set from dad of the same chromosome, say it's chromosome number 7. Those next to each other, homologous chromosomes. Which label identifies a 5' end of this DNA molecule? And this is a single nucleotide. The answer is A. If we look at the sugar in the center, there's an oxygen at the top of the pentose, so the pentagram shape made by the sugar. And you count clockwise the number of carbons. So that first one that B is pointing to, that's 1', then 2', 3', 4', and sticking off the little stick, you see CH2, that is the 5' carbon of your nucleotide. What type of bond is inside this red box? Another way to ask is, what type of bond holds nitrogenous bases together? It is a hydrogen bond. The sugar backbone on the two sides indicated by A and C, those are held together with phosphodiester bonds. Those are really strong. But the nitrogenous bases, the A and T and the C and the G, that bond is just a hydrogen bond, which is nice and weak because you want to be able to break it and put it back together. Hydrogen bonds are like Velcro. So you want to be able to pull apart your two strands of DNA and then let them go back together. Which label is on the leading strand? We're looking for A, B, or C. They're in red. DNA polymerase 3, which is labeled N, is pointing toward the replication fork, and it is continually synthesizing the new piece of DNA. So that means that strand A is the leading strand. Strand B on the bottom, you can see ligase and primase working on it. You can see the Okazaki fragments. So that indicates to us that this is the lagging strand. What is R identifying? R is right in the reproduction fork. That's right, yeah, reproduction fork. And it is taking the double strand of DNA and unzipping it, or breaking the hydrogen bonds between the two original strands, which means that R must be helicase. N is synthesizing new DNA continuously. What is N? N must be DNA polymerase 3. Which of these is the least likely to be shown in this picture? DNA polymerase 2 is not shown in this image. And that's because DNA polymerase 2 is an editor. It wants to fix errors in replication, but first you have to do the replication. A through D, all of those four options are visible on this diagram. So if you don't know where those are on the diagram, take a moment and find them, but each four of those do show up. Which comes first during the process of protein synthesis? Transcription. I ask this because sometimes we get the process of DNA replication and protein synthesis confused. And so some people will answer A, you have to replicate your DNA before you can make a protein. And those processes are separate. So we're going to replicate our DNA anytime we're going to do mitosis or cell division or meiosis, but we make proteins constantly, even in cells that don't divide very often, like neurons or fat cells. So DNA replication, protein synthesis, separate process. Protein synthesis has two steps: transcription, translation, and transcription occurs first. Which process begins with an RNA molecule and produces a chain of amino acids? Translation. You are translating the mRNA into a protein. Which cellular process involves the ribosome? Translation. Translation is using the mRNA, bring it to a ribosome, and the ribosome synthesizes the protein. A single-stranded molecule that contains the base uracil is produced during which cellular process? mRNA is a single-stranded molecule that contains uracil, and it is produced during transcription. Amino acids are carried to the ribosome by tRNAs. Little t means transfer, so they're transferring the amino acids over to the ribosome. You have identified a new protein in Drosophila, which are fruit flies. After extensive research, you have determined that the protein is 333 amino acids in length. How long is the protein coding portion of the mRNA for this protein? So let's do a little bit of backward math. Each amino acid is coded for by a codon. A codon is made of 3 nucleotides. So you need to take your number of amino acids times 3, and that will give you how long in nucleotides the DNA coding portion of the mRNA is. So 333 times 3 would be 999 nucleotides. If the template strand of a DNA molecule has the sequence 5' G, T, A, C, G, T, 3', what is the sequence of the non-template, the coding strand of DNA? We know that our DNA is anti-parallel, goes in opposite directions. So the coding strand must start with 3'. So we can already go ahead and get rid of option A. Then we look for the one that matches the DNA code we've been given. So it should be 3', C, A, and then we have B and D. The difference between those two is one has a T and one has a U. But we're looking for the strand of DNA, so we want to use the one with a T. That means that the answer is D. What amino acids do AAA and GGA code for? So you use this table by finding the first letter, which is A, that's on the left side, see the left title. Then the second position, which is A, so you go sideways from the A and then down from the A, and that brings you to this box where the top codon is AAU and the bottom codon is AAG. You can see that in that box, it shows you AAA codes for lysine, so LYS. Two of our options have lysine, so it's either A or C. And now let's do the same thing for GGA. The answer is A, lysine and glycine. From which of these regions would protein be more likely to be produced? A, euchromatin. U means true, so it's like it's the one that you're going to use. It's euchromatin, it's more likely to be used. In the rat licking experiment, the stress response of well-licked and under-licked babies was examined. What is the dependent variable in this experiment? And you can figure it out from the information given. The dependent variable is the thing that we are measuring, the thing we are examining. So that's going to be the stress response. The independent variable is what you're changing. That would be the amount of licking. A mutation that causes a single nucleotide switch but does not change the amino acid is called silent. A mutation that causes a codon to change from UUA to UAA is called nonsense. So a silent mutation does not change the amino acid. That would be if we did UUU change to UUG, it would still code for leucine. In a missense, you can truncate your amino acid, you can change the amino acid, and that might change the function of your protein. And in nonsense, you're going to truncate the amino acid by inserting a stop codon. UAA is a stop codon. Remember, we have our shortcut for numbering stop codons. It's you are awful, you are gross, and you go away. UAA, you are awful. Koalas have 16 chromosomes in their somatic cells. How many chromosomes would be in a koala cell after the completion of mitosis? (16) Mitosis creates identical cells to identical daughter cells. A somatic cell is any cell except for sperm and egg. So they're just body cells. Those are the ones that do mitosis. A Tasmanian devil gamete contains 7 chromosomes. How many chromosomes are in a Tasmanian devil skin cell? 14. Gametes are haploid, skin cells are diploid. You times 7 by 2, you get 14. During what step of meiosis do sister chromatids line up on the metaphase plate? Well, they're lining up on the metaphase plate, so it has to be either metaphase 1 or 2. In metaphase 1, the homologous pairs line up. In metaphase 2, the sister chromatids line up. So the answer is E. A white chinchilla is crossed with a gray chinchilla. All of the offspring are white. What does this tell you about the white allele in chinchillas? White is the dominant trait in chinchillas. 2 gray chinchillas mate and have pups. What is the chance that they will have a white pup if white is the dominant trait? Zero. 2 gray chinchillas must be homozygous recessive, so they do not have a dominant allele to pass on to their children. How is this trait inherited? This is an autosomal recessive trait. I can tell that it's recessive because down in the last generation, the one with the afflicted male and the afflicted female, those children that have this trait came from parents who did not. So that's generally only showing up in recessive traits, because if you have 2 heterozygous parents, you can produce a child that's homozygous recessive. If this was a dominant trait, more folks would have it. And if the child has it, that means that the parent also has to have it. So whenever you see 2 parents without the trait produce a child with the trait, you know that it's going to be recessive. What is the genotype of the individual indicated by the red pointer? That male does not have the trait. The trait is recessive. So we know that that male must have at least 1 capital A. Because that male's mother is homozygous recessive, she could only pass on a little a to that child. So that child, that male, must be heterozygous. How is this trait inherited? It's only males that are being affected, so that tells me that this is a sex-linked trait. And down on generation 4, individual 4, that afflicted male came from parents that did not have the trait being expressed. That tells me that this must be recessive. So this is D, a sex-linked recessive trait. What is the genotype of this individual indicated by the red arrow? We know this is a sex-linked trait, so we do need to include the X's and Y's. That's the only time we use the X's and Y's. That's why we didn't do it for the last pedigree. This is a square, which indicates that this person is male. So we're going to give that person a Y chromosome. So now we're narrowed down to D or E, and this person is not expressing the trait. So the answer is going to be D. This is a male with a, E. This is a male with a dominant allele because he's not expressing the trait. If we look over at the person labeled number 1 in row 2, the male that is afflicted, that would be D. That's a male with the trait. So the answer is E. Mary has blood type O, and she has a baby with blood type O. The father must also have type O blood because O alleles are recessive. Is that true because only O-blooded people can have O alleles? Or is that false because the father can have any blood except for type A, B? So the question was, Mary has type O, the baby has type O. Does the father also have to have type O? And that is false. The father does not have to have type O blood. The father could have A blood or B blood, but their second allele would be a little I. That means that they could pass on that little I to their offspring, producing an O-type baby, even though they could be A or B. That would not be true if the father was AB blood, because AB blood have A and B alleles, so that individual would not have a little I to pass on to the baby. He could not be the father of that baby. All right, so I hope that was useful. I hope it was fun. If there's any ones that you're totally stumped on or confused about, please email me. I'd love to talk about them, and I hope you have a great rest of your day.